For our next session, it's John Lauerman, who is a health reporter, and he was covering personal genomics and became the subject of his own story, and he's going to tell you about that. And I also would like to encourage you to go look at his collection of articles at Bloomberg called The DNA Conundrum. John?
Hi, it's a pleasure to be here. So, about 11 years ago, actually 12 years ago, I was on a press tour of the Whitehead Institute, and Eric Lander, who I'm sure most of you know or have heard of, was leading the tour, and he was showing us all this fantastic and futuristic and now outdated technology that was being used for the Human Genome Project. And so he's leading us around Whitehead. And at one point he happened to mention that most of the DNA being used for the project was from one volunteer from Buffalo. And I'm from Buffalo, so I immediately felt a kinship with this person, whoever that was. And I also started to think, this sounds kind of interesting. Sounds like something, that perhaps I could do. Not too many people were getting sequenced in those days. And then I happened to bump into this guy a few years later, who I think most of you know. And so we started talking about this future in which everybody was going to get their genome sequenced. And I just found that fascinating. Sound like Star Trek. Scotty, beam me up, and while you're at it, sequence my genome. So I thought, why wait for the future? Let's do it now, and maybe even write something about it. So a couple of years ago, I had just finished up a series of stories on education on for-profit colleges, and my editors were very excited and wanted to know what I wanted to do next. And they asked me what I had planned, and I said, well, this is what I'd like to do. I'd like to join the Personal Genome Project and write about it. So my editors were very excited. And the first thing they did, a bunch of people I work with used to work at the Wall Street Journal. The first thing they did was they pointed me to this story here. It was written by a pretty relatively healthy guy, otherwise healthy guy like me, who one day in the course of researching a story on longevity discovered that he had a malformation in his blood vessels that put him at risk for an aneurysm. And so he ended up doing this Pulitzer-winning piece on aneurysm. And my editors were asking me, do you think there's any possibility that anything like this could happen to you? And they sounded almost a little hopeful, actually. They sounded very hopeful. And I said, I've spoken to a lot of people who have had their genome sequenced, and I really don't think there's much chance of that, but I suppose it could happen. So I said, most of the people I've spoken with have pretty boring genomes. So I decided to go ahead with it. Really, this is the motivation behind this has more to do with this quote here. One of my colleagues at work was formerly posted in China, and he has this quote framed on the wall behind him. And it's an exchange between Samuel Johnson and his biographer, James Boswell. And Johnson had said something about wanting to see the Great Wall of China. And Boswell says, I'd love to see the Great Wall too, but I have kids at home, I'm kind of a busy guy. And Johnson replies to him that seeing the Great Wall be of great importance in raising your children to eminence. There would be a luster reflected upon them from your spirit and curiosity. They would be at all times regarded as the children of a man who had gone to view the Wall of China. And for me, having my genome sequenced, that became my Great Wall. So I did everything that you were supposed to do. Everybody's, I think our colleagues from Canada here talked about the requirement to speak to your family members. I don't know if I was required to do that by the PGP, but in any case, I did it. I talked with my wife, my children, my brothers and sisters, my parents, and they were all in favor. I decided to go ahead. And the thing was, in my mind, it was never intended to be diagnostic. I gave blood and saliva samples for the study right in George's office. And I sort of sat back, and for a few months, actually longer, it took longer than I thought it would. Nothing happened. At one point, actually, I called George late at night. My editors were saying, where's the story? Where's the genome story? And George had to sort of talk me down off a ledge. This is oops, I think I have the wrong slide here. I was going to show you a picture of my genome being sequenced at Complete Genomics in Mountain View. I wonder if this takes me back. Well, anyway. That's because this is a picture I want. So this is my genome being sequenced at Complete Genomics in Mountain View, California. And I'm sure you all know, Complete Genomics was recently bought by BGI. So I nearly had made in China stamped on my genome. I finally did get the results January 2nd. It was a holiday. It was late in the day. I was sitting alone in my office, and I got an email actually from Madeline Ball saying, you should be getting your results in a few minutes. And so this is what happened next. Not too long after I got my results, I was speaking with this gentleman. His name is Mark Sanders. He's a former firefighter and a champion weightlifter that I interviewed. And the reason I talked with him is that we have a few things in common. We're the same age. We were both born in Indiana, where he still lives, and we both have a rare mutation called JAK2 V617F. JAK2 V617F is everybody calls it JAK2 actually. What it does is it sort of revs up replication in bone marrow cells that give rise to platelets and red blood cells and other things. I'd never heard of this mutation before the PGP told me about it. It's actually associated with a set of disorders called myeloproliferative neoplasms. It doesn't exactly trip off the tongue. They're described as cancer-like because they're associated with these high blood cell levels. Mark had one of the most dangerous disorders associated with the JAK2 mutation. He has myelofibrosis. He stopped making red blood cells completely, and he had to have a bone marrow transplant, and he almost died. We had a long talk about this, and after I got off the phone with him, the next question on my mind was, would I develop the same disorder? I don't think we know enough about JAK2 mutations to say whether I will or I won't. But what I did find out was that I have another relatively rare condition called thrombocytosis that has given me high platelet counts that increase clotting. And actually, what I discovered was that these were associated with a symptom that I'd been having for years called scotoma. And scotomas are small blind spots in your vision. Nobody had ever sort of put all this together. I'd had high platelet counts for years, and I'd actually been to the emergency room, had my blood taken on more than one occasion. And nobody ever sort of put two and two together. People tend to get high platelets when they're sick. So people saw me with high platelets. You know, you have something else going on here. That's probably why you have a high platelet count at the time. So if I hadn't had my genome sequenced, there's every possibility that I would have gone for years, maybe till now, without having been diagnosed with this disorder. So I continue to write about this topic. I think there's obviously, you know, from the discussions, there's any number of topics related to genome sequencing that are interesting to reporters. This is Camilla Grandal and her daughter. This woman and her daughter, her family live in Utah. Both of them underwent sequencing to find a gene that was killing baby boys in the Grandal family, usually by their first birthday. So scientists found the mutant gene on the X chromosome, and when Camilla became pregnant with a boy, she wanted to know if she had the gene that caused the disorder. Researchers at the time were still struggling, and I think are still struggling, with what to do when it's appropriate to tell patients about what's been found in their genomes, particularly when those genomes have been analyzed for research purposes. And by the time these scientists that worked on this project that Camilla was involved in were able to tell her whether that she had the mutation, her baby boy was already dead. This is the Christman family. This guy Matt Christman, the sort of heavyset gentleman who's in the foreground, has hypertrophic cardiomyopathy. His insurer, WellPoint, wouldn't pay for a relatively inexpensive test, really only costs a few $100 for each of the kids, and I think about $1,000 for him. Wouldn't test him for this gene. They said that the test wasn't effective. After we wrote a story about him, WellPoint offered to pay, but another company called GeneDx had offered to do the testing for free. And it turned out that 5 of his 7 kids were affected, so 5 of these kids were at risk for the same disorder, and were walking around effectively undiagnosed. And it's my opinion that people shouldn't have to go to the media to get these tests paid for. In December, I went with my wife Judy. We went to the Faroe Islands, where they're starting a project to sequence the genomes of everyone in the country and link the results to medical files. There are still some people there who are concerned about how the information will be used. Again, it's becoming routine. It goes without saying that some genomes will produce unexpected results like mine, and that everyone will have to figure out at that point what to do about it. This is Melissa Butters and her son Patrick. This is one of the terrific stories out there about genomes. Melissa had a rare growth disorder, really a cancer disorder, that she grew up with, and after her son was born, the 2 of them had their genome sequenced, and they figured out exactly what the disorder was and what gene was causing it. There's no treatment for it yet, but as a result of piecing together these pieces of the genome, eventually there may be. I've been, down this road myself. I thought I had one of the most expensive diagnostic tests out there. Now George says today that they were only paying $2,000 in 2010. So it's not even close to being an expensive diagnostic. I discovered some things about myself that I maybe wasn't prepared for, but as it turns out, now I'm taking an aspirin every day. I don't see scotomas anymore. I have no other symptoms, at least right now, from thrombocytosis. It's worked out remarkably well for me. I think that it's very important, obviously, to figure out some of the answers to some of these other questions that we've talked about. But I'm extremely grateful to the PGP. I'm glad I joined, and I'm happy to take any questions. So.
Hi, I have a question.
Sure.
How was it speaking with your family about participation in the program?
So that was remarkably easy. My family is kind of used to, in my life as a journalist, just taking on strange projects, sometimes becoming personally involved in them. I think probably the most difficult part of it was talking to my children about it, because I think they were least capable of, I guess, understanding what it was I was volunteering for, and most likely to go along with it just based on their, interest in cooperating with me. So we had a lot of long talks about it, and but I think overall it was a good discussion. Actually, I think I was told that I could only take one question, so if anybody else has questions, I'd be happy to take anything further later on. So thanks again for your time, and it's a pleasure to be here.